Hello and welcome to Greiner Bio-One Talks. My name is Anna Maria Šimundić and I am a Global Director for Medical and Clinical Affairs at Greiner Bio-One. We are here today to talk about a very challenging topic in preanalytics, and that is hemolysis. And we will be talking about some current trends, but also some future insights and future challenges. And I have a very special guest with me here today, and that is Gian Luca Salvagno. Welcome, Gian Luca.
Thanks, Anna Maria. Thanks very much.
Gian Luca is a full professor at the University of Verona, and also a director of clinical laboratory services at Peschiera del Garda Hospital. So welcome once again, and thank you for accepting our invitation to talk about this topic. I think that hemolysis is a holy grail of our in laboratory medicine.
Absolutely.
Yeah, it has been and is still a greatest challenge for laboratory professionals.
Exactly.
So can we start with some general, definitions? What is hemolysis? How do we define it? What are the causes? What are the consequences? And what are the requirements from, regulatory requirements, ISO? What should we, as laboratory professionals, be aware of when thinking about a hemolyzed sample?
Sure, Anna. Yes, hemolysis absolutely is one of the main important source of errors in laboratory medicine. The meaning of hemolysis, we have erythrocytes, and for several reasons, erythrocytes are broken. Hemolysis, the meaning is the lysis of erythrocytes. Why? We have two important categories. One is in the body fluid, a disease, but there are very few percent. And the other case, we have most causes of hemolysis are preanalytical phase during the phlebotomy. We have several diseases. We have antibody against erythrocyte that destroy the erythrocyte, and this is in vivo hemolysis. But it is very, very few percent, less than 5%, 3%. The most case of hemolysis that we found in clinical laboratory are due to during the procedures, during phlebotomy collection. Why is a problem? Because when erythrocytes are broken, we have all inside erythrocytes that go outside. We have some kind of substance, for example, potassium, LDH. We have enzyme, for example, we have protease inside the erythrocyte that destroy completely some protein. For example, insulin. It's not possible to measure insulin in hemolyzed sample. We have problem because we are inside the erythrocyte, we have folate, for example, and when the folate go outside, we're not able to measure the real amount in the plasma. But there is another question, is about the color. Because inside we have hemoglobin, red color, the plasma change the color from yellow to red. And when we move this the modification, we have influence in several analytical way. For that reason, for clinical laboratories, a real huge problem when we receive a sample, we have a different level of hemolysis. For that reason, 20, 30 years ago, our European and international society organize a meeting and new recommendation to detect hemolysis, because it's important to know if it's a real hemolysis due to in vivo pathological disease or if it's a hemolysis due to a incorrect sampling, especially during when you use several kind of needles or different kind of tube.
So how do we, I think it's very important what you said, that laboratories need to detect hemolysis.
Absolutely.
Because, so what are the ways to detect hemolysis, and what would be the most recommended?
In the past, we used by eye, visual inspection, but there is a lot of problems. There is a qualitative measure. And around 20 years ago, we started to use instruments that are able to measure exactly the amount of hemoglobin, of the free hemoglobin that we have. Mm-hmm. We have a lot of scale that we are able to evaluate with this scale. Is a spectrophotometric analysis. And then we have to able to evaluate the total amount of free hemoglobin. In that case, FLM and other international society develop a recommendation how to report the results, because in some case the sample we cannot analyze the sample because there is too much hemolysis. This evaluation is not quite, it is quantitative. If the sample have high level hemolysis, we cannot perform a lot of tests, of all tests. If you have a middle range of hemolysis, we can perform some kind of test. If you have low level of hemolysis, we can perform more tests. Another topic is when we report the results, because sometimes we know that the results we have a little amount of increase or decrease of the concentration due to hemolysis. And for that reason, we have a lot of discussion in our society, and there is no final decision, but the most nowadays, most of the colleagues decide to report if we are inside the reference change value of the substance. What is the meaning? Is that we know that there is an increase due to hemolysis, but this increase is not so high to not to give a report, because also we have to give information to the patient, because hemolysis is the principal causes of recollect sample. And when we correct sample, we have a free perspective, because we have delay in the diagnosis and treatment. We have problem in the organization, because we have to recollect from the nurse the tube, new blood sample, and we have also economic perspective, because we have to spend time. Time is really important. Time of the medical doctor, time of the nurse, time of the clinical laboratory. For that reason, there is a consensus document in which there is a recommendation for clinical laboratory that in some case you can report some part of the results, but in which you can specify inside the report that there is a low level of hemolysis. Nowadays in laboratory information system we have all this information, because in clinical laboratory, especially in clinical chemistry, in coagulation, immunochemistry, we perform hemolysis for all samples. We start a few years ago. Nowadays, a lot of most of the clinical laboratory prefer to analyze all samples for hemolysis, because we have also the problem is the low level hemolysis. Because for some kind of test, for example, LDH or AST, is enough a very, very low level hemolysis to increase. LDH is important biomarker for oncology, especially for hematology, and for that reason there is more attention.
Let us maybe go back to detection, because I think that there are still some issues that I would like to discuss. So would you agree with me that currently the way we detect hemolysis using different equipment, different instruments, is not fully standardized?
Yeah.
Right? So there are differences in the way different analyzers detect. So the method of detection, wavelength they use. Some are, as you said, qualitative, some are quantitative. So could you comment on that? And how do you see, the way forward?
Yes, so we have seen that most manufacturers have a semi-quantitative scale. They have not, they perform 0, 1, usually 6, 7 blocks. Some manufacturers are able to perform a continuum scale in which there is a quantitative evaluation hemolysis. Sure, several years ago, we start to work to the quality control of hemolysis index. In clinical laboratory nowadays, we have a system that check how we measure hemolysis, is quality control. And with quality control, we are able to evaluate if how our system is able to check hemolysis. Should we need to go in the direction of standardization also for the test, because hemolysis index is a clinical laboratory test, not reported in the report, but is absolutely a test such as glycemia, LDH, and other. And we need standardization, and we are working on standardization because in the different equipment we perform hemolysis index in the clinical chemistry instruments, and this test is not, until now, unfortunately, is not so standardized. This is a huge problem that it takes the society, the company to work together to perform a standardization, to have the scale, quality control, in which you are able to evaluate this test, and also external quality assessment. Another important topic is the proficient testing, external quality assessment for hemolysis.
So let's talk a little bit more about detection. So one thing that is also very important are the decision limits for hemolyzed samples. So could you comment on that? What is there for laboratories to use? Obviously, there are some decision limits that are recommended by the manufacturer. Is that the way to go? Should laboratories verify and produce their own decision limits? How do you see that? And what is the case in your laboratory? How do you do it in your lab?
Yes, correct. Manufacturer in the data sheet give us information about the interference by hemolysis, but there is one issue inside, because sometimes the manufacturer put only hemoglobin and not a lysis of erythrocytes.
Yeah.
This is quite important, because sometimes we have not interference by free hemoglobin, but we have interference due to the cytoplasma of erythrocyte broken.
Cell components.
Exactly,
Yeah.
perfect. For that reason, what we have done in our lab, we have performed, according FLM, we have performed an evaluation about the interference of hemolysis. We perform some experiments that are published by our group in Italy and our group international, describe where is this influence, and to decide the cutoff, the limit of decision limit. When we decide the decision limit, we have a three level. White level is okay, the hemolysis is below a cutoff. We have the red limit, the highest, where is too high the level of hemolysis, we cannot put the results. The question is about in the intermediate, because in that area we decide to report the results of the patients, but we have also a claim inside. There is a sentence in which we describe that there could be an interference due to a hemolysis. We don't like to use a formula or other to correct. We prefer to report the results and give this information. Sometimes, for example, I think for the children, for the pediatrics, it's important to have information instead if there is a grade of hemolysis. Why? Because the repetition of the sample, it takes a lot of time, since it's absolutely difficult to have a new blood sample. For that reason, we report both the comment and also the final value. We are use the FLM recommendation and also the FLM table about this.
I really like your position on providing factoring or calculated result, and I fully agree with you. There is no, in my opinion, that is not acceptable. But could you comment on that? Why is that not acceptable?
In the past,
because there are laboratories
in the past, our colleagues tried to develop, because they want to give a result, and they are positive because they think that is possible by using some kind of mathematical formula correct this value. The question is about that in biological system it's difficult to use this formula to exactly describe, give the information. The meaning is, if because I have this level of hemolysis, my real sample should be have another value. But this is purely theoretically, because in the biological system it's difficult to perform this form to everybody, to our blood, because there is a lot of difference between there is the inter-individual variability. For that reason, and also with the statement of FLM, we don't like to use the formula. We prefer to use a scale and to report the result when is possible by using this cutoff.
So what is the ISO requirement in terms of hemolysis?
ISO perform a lot of documents, and we need to know the interference, because hemolysis is an important huge interference, as ISO and other important recommendation says that clinical laboratory should know the influence of hemolysis on the sample testing.
Yeah.
And also describe that the clinical laboratory should have standard operating procedures written to check hemolysis, how to check hemolysis, how to report hemolysis. And also we have these from ISO and from the accreditation program, especially the 15189, that the question of interference clinical laboratory should learn and should also show to our physician or our patients.
Okay, so let's move now to understanding the causes of hemolysis and preventing hemolysis. So my question to you would be, maybe we start with the general note. What are the major contributors to hemolysis in your opinion? How do you suggest, one identifies these causes, and then how to address them?
In vivo hemolysis is very, very few cases. More than 90% of the causes are due to preanalytical phase. And in preanalytical phase, the more important aspect is during the blood collection. We know that from the literature that the needle, the size of the needle is a source of errors, a source of hemolysis. When we perform also the vein, because if we use, for example, the vein of the hand, we have a high level of hemolysis instead the vein of the arm, because it's due to the mechanical stress of the erythrocytes when we take the erythrocyte from the vein until the blood tube. Another question is about when we have this most important source, because when you perform blood collection and you see the blood go inside the tube, perhaps drop by drop, because it's a difficult vein, in that case we have incredible increase of hemolysis. Unfortunately, hemolysis we have not in patients that perform blood collection one time each year, but for patients, for in the ecology department, for patients that have a lot of drugs, that have a lot of treatment. For that reason is the principal source is during blood collection, and we have to aware the nurse and the people that work in the blood collection service the principal causes, and we have to find some solution. How? By using not one needle, by using what is necessary. And for my opinion, is really important that the nurse have all information, everything is known, and also the instruments, because sometimes for one patient it's better to use one needle instead another needle, a butterfly instead a direct needle, because in my opinion, is important that the nurse have all instrument that for her is necessary to perform the best blood collection. And in that case, we try to reduce. Unfortunately, we have the problem with some kind of vein, and then in some cases it's very difficult to reduce hemolysis. But for me, the first point is education, and second, to have a high quality level of preanalytical instruments. Needle, order, butterfly, quality material, because also an interesting aspect is the needle, because the needle is not always the same, not only about the gauge, but also how the needle are performed inside. Because there is incredible the technology to produce a needle. Around the world, there are a lot of companies that produce needle, but there is a lot of technology. And if the nurse have the best instrument that we have, we can reduce the level of hemolysis. There is another, we know that we have a high level of hemolysis not in outpatient, but in inpatient. Why? Because in the outpatient, we have expert phlebotomy service. And this is the reason that we have low level. For inpatient or in emergency setting, we have a lot of people that perform, and for that reason, we need to work more on education and to try to give to the people the best instrument to perform. Sometimes they don't use direct butterfly, but they use other systems that are just in the patient, but that we use for the drug, not to collect the sample. And this is an important source of hemolysis.
So let's maybe try to dive into more details. So needles are important, obviously, and you were talking about the patient veins, and I guess it's the entire system that matters. You know, the needle diameter, the vein, the pressure in the vein, and the pressure in the tube. So how it relates to hemolysis? Is it better to use high diameter needle or smaller needle with smaller diameter? Is it so? How, if you can explain the physics of that system, that would be great. How it relates to hemolysis?
First of all, you start with the vein. What is the bed is the vein of the arm, and if you don't find nothing, you go in the vein of the hand, because here is the best to perform the vein. Second, the tourniquet. We cannot use the tourniquet more than 1 minute, we know. And because the tourniquet is able just to check the vein and after that you put away. Tourniquet is another source of hemolysis. Second, we have absolutely the needle, but, and not only the needle, because also we have the butterfly, because for the butterfly have a higher level instead of direct needle. Because we have the tube, and inside the tube, the erythrocytes can have some mechanical stress. Finally, we have when the sample arrives in the tube, the pressure, because in the vacuum tube, the erythrocytes go very fast. But if you use blood tube with low volume, there is less pressure, and we have low level of hemolysis. For that reason, we need to know all aspects to reduce hemolysis. And and when we finish the blood collection, we have other aspects. We take the sample and put the sample to, for example, in the clinical laboratory. If you use pneumatic tube, for example, pneumatic tube is a source of hemolysis, because the tube inside, there is some mechanical stress. Mechanical stress for erythrocyte should be just when we mix the sample, mix it gently. But if we mix it very strong, we have increase of hemolysis. Pneumatic tube, we have to now quite indicator of pneumatic tube, because pneumatic tube is a source of hemolysis. The temperature, the time, because also the sample should be arriving in the clinical laboratory within 1-2 hours. But if it's delay, there is some source of problems. The question is about that the principal source is blood collection, but we have a lot of other area where we have increase of hemolysis. In this moment, the pneumatic tube, I think, is one of the most topic.
So you were saying that the pressure within the tube may cause hemolysis. So my question then to you is then the use of low draw tubes, so tubes with lower volume of lower pressure, is that the way to minimize hemolysis?
For the erythrocytes, when the erythrocytes arrive in the tube, if there is a low pressure, there is a low mechanical stress. There is some kind of paper that describes this, but we can perform, if you, for example, some study, because theoretically, yes, because when we have other system, for example, the syringe, we have low level of hemolysis because we take slowly the blood inside the syringe. We have some kind of manufacturer that perform different kind of level. The vacuum tube we have to use with attention because could be a source of hemolysis.
Things that you mentioned are actually known for quite some time, but somehow hemolysis still remains a challenge. Even though we do understand the theory, the needle, the tube, the tourniquet, everything you described, it still remains the challenge, right? It's still, so that is not resolved. So my question to you would then be, do you see maybe some innovation that would be necessary or that we would need to address this problem of hemolysis?
Yes, it's absolutely is a problem 40 years ago and until now. I think that by watch the result for the outpatient, we have seen that with expert phlebotomy, you are able to reduce because in the outpatient service, we have low level, very low level. We need to work more in the emergency department. And here we can work with absolutely education. And secondly, we can to work more in the needle, in the kind of needle, because when we use a very, very low size of needle, we have increased amount of hemolysis, and we can use those to work more in the connection between the needle to the tube and also to the tube. Research continue to work because we know that we have research in vacuum tube to try to reduce the mechanical stress on the erythrocyte, because the question is to reduce the mechanical stress.
Yeah.
In the same the pneumatic tube, because in pneumatic tube nowadays in the tube the sample go very, very slowly, because the problem is that if it is too high speed, we have
mechanical stress.
Mechanical. The question is about to work in research in the phlebotomy equipment to reduce the mechanical stress or erythrocyte from the beginning, from the vein until the tube.
So, what would be, in your opinion, then the way forward? What do we need to concentrate on? Each individual, laboratories, professional organizations, manufacturers, everybody has a role. So what do you think is the way forward for each stakeholder?
I work in the university, and in my opinion, nowadays we need to work a lot in the education, in the course, workshop, especially for the people that perform phlebotomies. I think that education nowadays is a first level, because sometimes phlebotomy we consider it is a very easy job. Easy, fast, don't take care, just one drop inside the tube. But I think that we need to put more attention about the education, training, and to have a certification. Okay, you are certified because you have performed sample with low level hemolysis. We need some kind of indicator about the people that perform phlebotomist. I think that for my point of view, for university work and for the professional category, work more on education, and for the hospital work on indicators, because for example, if I have more high level hemolysis in the morning and not in the afternoon, or vice versa, or if we have hemolysis when we have some kind of people and not others, we can try to, we have to perform education and training.
Yeah.
Because the people that work in the hospital know that sometimes the hemolysis in the amount of hemolysis tube change depend on who were in the emergency that perform, because it's experience. We have a lot Well, people that high level of experience, people with low level experience. We have to evaluate. We need the indicator. We need to report. We need to discuss together, and we need to check from where the people and to perform some kind of course. For the manufacturer, obviously, we need certification, scientific evidence that the product are exactly what we want. We need scientific literature, and we need that research. And research meaning we need that the company try to continue the work in the different part, material, but also in the engineering, because there is a lot of engineering inside a needle, inside a tube, inside whatever take care.
Yeah, you're absolutely right. And I think each stakeholder that you just described, so individuals, specialists in laboratory medicine, but also everybody involved in phlebotomy or blood collection, laboratories, professional organizations, manufacturers, everybody has a role. And the final goal, I think we always need to keep in mind that the final goal is actually to do what's best for the patient, right? To minimize the amount of errors, to minimize the risk for patient, and to increase the quality of the test results. So this is my last question to you. You have a long career, obviously, and you have in your career you have met a number of interesting cases and situations. What would be some most memorable moment in your career, pre-analytical moment, where something that you did meant something in a patient perspective?
I remember just in this moment, because we have performed blood collection for the patients. We have a glucose after glucose. Sometimes the patient take a glucose test, and after that we perform glucose and insulin for 6.4 points. And I remember that for the glucose it's okay, but for insulin I have absolutely strange results. Incredible strange results. Technician called me, strange result, and we start to think about there is something wrong about the timing, because the curve of the insulin is very strange. And we have thought that the nurse have wrong about the timing. I don't understand. Finally, the nurse says me, no, it's perfect. I am sure. There is also the minute on the lab. I'm sure. And finally, take my technician, okay, watch the sample. And I watch the sample, I have see low level of hemolysis in one tube. The insulin decrease due to hemolysis. In that case, I have says, okay, because we have hemolysis, not all sample. In clinical insulin is an immunochemical test. And I say, this is the question. I remember because at the beginning we have said, okay, the nurse performed some mistake, but the problem is about hemolysis inside the sample, not correctly detected, and we really understood that we have a problem. And then we have resolved this question because we talk with physician that in that tube, in that we have low level of insulin due to high hemolysis, because this is one problem that we have.
Yeah, this is why I think personal touch will never be replaced by an instrument or an AI, because a person that has all this knowledge is able to understand and solve such complex problems. Thank you, Gian Luca, for being my guest and for such exciting discussion about hemolysis. I really hope that you have enjoyed this podcast as much as I have.